ladies and gentlemen. I think we should really start now. We have 7 exciting talks coming up in the next 2 hours. I'd like to ask all the speakers to stick to 12 minutes roughly, so that we have 3 minutes for discussions and questions immediately after the end of the talks. And if time permits, we can have a general discussion after that. We'll start the session with Dailey from King's College in London, and she'll be delivering a lecture on the biodistribution of inhaled nanoparticles, visualized using SPECT imaging. Leanne, please.
Okay, thank you very much. I'd like to thank all of you for getting up so early to come to this session, or for those of you who stayed out all night to stay for this session. And I'd like to talk a little bit about our work that started with a very clinical or clinically relevant question. So my interest in inhaled nanomedicines started back when we were working with clinicians who were involved in the development of inhaled iloprost to treat pulmonary hypertension. And one of the problems with this therapy was the fact that the drug itself has a very, very short half-life in the lung, which means that patients have to take their medication 6 to 9 times daily. At the time that this product was actually developed, the inhaler technology was quite bulky, so it made it very, very difficult for patients to actually have a certain standard of quality of life. Inhaler technology is now improved, but the fact remains that they still have to take their inhalation therapy quite often during the day. So wouldn't it be wonderful if we could use an inhaled nanomedicine to actually achieve a once or twice daily administration rate by slowly releasing the drug compound over time in the lung over a period of 12 to 24 hours, which is where my interest in inhaled nanomedicines really began. Now, at the time, we, like many other groups, were very naive. We focused quite a lot on the formulation development, trying to get the drug into the nanomedicines, trying to get the drug out of the nanomedicines at the right time, and the right pharmacokinetics. But really, if you're thinking about the pharmacokinetic profile of a nanomedicine, you have to think about the particokinetic profile of the nanoparticle, as well as the drug release profile. And so really, over the past few years, my group has been trying to investigate particokinetic profiles from the lung. We focus on biomaterials primarily, so we're looking at a wide variety of different types of nanoformulations. These can include polymers or biopolymers, but we also focus a lot on protein-based particles using albumin, as well as lipid particles, solid lipid nanoparticles, lipid nanocapsules. Our idea is that it's important to actually look at a wide range of formulations to, first of all, try to assess their biocompatibility in the lung. It's well known and well established that inhalation of certain types of nanomaterials can actually cause irritation and pro-inflammatory effects. So we've spent a lot of time screening different types of nanomaterials to see if we can find those which do not seem to cause an irritant effect. In the first instance, we've used a single dose administration study just to look at acute pulmonary inflammation. And from this study, we've been able to see that certain types of nanomaterials, such as hydrophobic polymeric nanoparticles, do act as irritants very quickly in the lung, even at therapeutically relevant doses. However, we've also identified a wide range of different types of materials, including protein and lipid-based systems, that seem to be well tolerated, at least at a single dose, and we can go up to fairly high concentrations, about 200 to 500 micrograms per mouse lung. So what we've done in this study is we've decided to take 2 of these systems, albumin-based nanoparticles and lipid nanocapsules. The albumin-based nanoparticles have a size of about 150 to 200 nanometers. They're cross-linked. And the lipid nanocapsules are about 50 nanometers in diameter. They consist of an oily core that's surrounded by a semi-solid phospholipid shell that contains a pegylated surfactant. So these are pegylated systems. We have covalently modified a linker, DTPA, to the surface of these nanoparticles. DTPA is a chelator for indium, and we used indium as our radioactive tracer for tracing both of these particulates in the lung. We administered the nanoparticles to the lungs of bulb C mice using an oropharyngeal aspiration technique. We use this technique because it's minimally invasive, and we found that if we had to use any other technique that involved injectable anesthetics, we had trouble waking the mice up during the imaging procedure or after. We have found that this dosing procedure actually gives you a fairly good distribution of the nanoparticles throughout the lung, and you reproducibly get a lung dose of about 40%. So if we just compare the two nanomaterials from first glance, you actually see quite striking differences. The lipid nanocapsules, which are smaller, actually show a very rapid or moderately rapid clearance profile. And you can see that the lung dose after 24 hours is substantially lower. In comparison, albumin nanoparticles have a very high lung dose that is maintained across at least a 48-hour period of our imaging studies. Let's take a look at that pharmacokinetic profile a little bit more closely. So I've taken the image analysis here, and we've normalized the values to 100% of the lung dose. And we found that albumin nanoparticles are cleared substantially slower than soluble albumin on its own. We've estimated a clearance half-life of about 96 hours just from these profiles. Soluble albumin has also a fairly long clearance half-life of about 27 hours, and we've all compared everything to a small molecular marker, that's indium DTPA, just to make sure that our label is not coming off and we're not seeing free label as a kinetic profile. If we compare this now to the lipid nanocapsules, which are smaller, we see a very, very different profile. We have a clearance half-life of about 10.5 hours. What we've done in addition in this study is we had the question of whether actually administering an inhaled medicine to an inflamed lung, which may be a part of the lung pathology of disease, would influence the clearance kinetics of the nanomedicine. So we pre-treated a subset of animals with LPS intranasally 24 hours prior to dosing the nanoparticles, and we had hypothesized that the inflammation would actually change the clearance profile. What was really interesting to see is that they were absolutely identical. So inflammation, either predating the treatment or let's say you were taking this medication for a non-inflammatory disease and you happened to have an acute respiratory infection on top of your treatment, this would not necessarily change the clearance profile of the nanomedicine. We were also interested to see what happens to the nanomedicines within the lung themselves, so we also carried out a subset of animal experiments which were terminal biodistribution studies. We took a lung lavage after each time point. I would like to say at this point that the image analysis and the terminal image or the terminal biodistribution studies were very, very close. So the SPECT CT imaging was in fact a very, very good indicator, non-invasive indicator of the biodistribution throughout the body. However, you can't really see what's happening in the lung with SPECT CT imaging. We saw that we could definitely characterize the nanomaterials in the lung lining fluid or the lavage fluid, and we saw that the concentrations were decreasing over the time period. We found that albumin nanoparticles and albumin solution were found in the lung lining fluid at much higher concentrations, and this is probably due to the slower release or slower clearance profile. Lipid nanocapsules disappeared quite quickly, and indium DTPA left the lung almost immediately. It was found in the bladder and was excreted. We were very interested to see if any of the nanomaterials were taken up by alveolar macrophages, so we looked at the cellular fraction, and we found that the albumin nanoparticles were really the only ones to show a moderate uptake, which peaked at about 24 hours and then seemed to disappear. Albumin solution was also taken up by macrophages, or found in the macrophage fraction, and that was slowly increased over time. The majority of albumin nanoparticles and soluble albumin was actually found in the lung tissue, so there was some adherence or partitioning, at least, of the albumin into the lung tissue. Lipid nanocapsules were almost not at all, very, very small fraction found in the cellular fraction of the alveolar macrophage population. We were curious to find out if these particulates were actually crossing the air-blood barrier and getting into the systemic circulation. Now, we have, unfortunately, with this technique, no ability to see if the nanoparticulates remain intact throughout this procedure. However, we could look at the signal of the tracer in the blood and all of the other organs. I'm not showing all of these, but I am showing the liver fraction because that can be indicative of particulate entry into the systemic circulation and then uptake into the liver. We did see that the lipid nanocapsule signature did increase in the liver over time, peaking at 24 hours, and then started to decrease. Albumin solution, interestingly enough, was increasing steadily. Albumin nanoparticles was very, very low liver signal, but it also was increasing slowly over time, and we feel that these profiles certainly are matching the lung clearance rates that we're seeing. So to summarize, we were thinking about all of these studies in a clinical context. What if you wanted to use these nanomedicines for treatment of certain types of diseases? Well, certainly albumin, due to its very, very long clearance half-life in the lung, may only be useful for therapeutic treatments where you have maybe a once-weekly dosing profile and one has to ask if this is going to be acceptable for patients. You don't want to certainly cause a lot of accumulation of nanoparticulate material, even if it is very biocompatible in the lungs. So we have to do a lot of multi-dosing studies to look at that. Lipid nanocapsules are kind of interesting because of this clearance half-life of 10.5 hours. They might be suitable for a once daily administration profile, and maybe we need to go back and revisit this for iloprost. And finally, I think what was really interesting was the fact that we saw that lung inflammation did not impact the clearance profile of the lipid nanocapsules, which was something that we weren't expecting. So I'd just like to thank all of my colleagues who helped participate in this study. Thank you very much for your attention.
Thank you, Mart. Thank you very much for this very nice talk. Questions from the audience? Yes, Avi, please.
Thanks for a great talk. Regarding the kinetics crossing the mucus on the lungs, could you maybe say what dictates that and what would actually affect that?
So you do get a deposition primarily in the central airway, so you'll probably have a lot of the nanomaterials caught in the mucus. We were interested in mucociliary clearance, but unfortunately, with this particular dosing technique, you get a lot of stomach deposition after administration. So in the first 24-hour period, we weren't able to measure any mucociliary clearance or distinguish it. However, after 24 hours, all of the dose that arrives in the stomach has been eliminated. So if any other stomach dose were to be identified in that period afterwards, that could be mucociliary clearance, and that is the case with albumin, but we didn't see any signal with the lipid nanocapsules. So yes, I think it's being trapped in the mucus. The lipid nanocapsules might possibly actually be able to penetrate mucus, but that's a different study. And so that's as far as we've gotten so far in that case. The rest that goes into the systemic circulation, we think must be the peripheral deposited dose.
More questions? Still have time for one or two. Yeah.
My question would go into the almost same direction, but I wanted to know, you mentioned that you pegulate the surface of your particles. So are you sure that you have the same amount of peg on both the comparators, lipid versus albumin particles, or maybe there's a variation causing this variation in clearance rate?
We don't pegulate the albumin nanoparticles. Those are purely albumin that's cross-linked, so we don't have any surface coating there. We found it was surprising that the lipid nanocapsules, the pegulated, actually cleared the lungs so quickly due to their size, and that especially soluble albumin does not clear, but we think albumin is highly regulated in terms of its concentrations in the lung, and there are many different uptake mechanisms for albumin in the lung. So we think the lipid nanocapsules, due to the pegulated surface, are avoiding, to a certain extent, some interactions with cells. They are possibly also maybe deformable, so maybe they can slip through the barriers, tight junctions in the lung, although we have no proof for this. So, and maybe they've also lost integrity in the lung. We're still investigating this.
Please.
Thank you for a very exciting presentation. You said that this method of administration can cause accumulation and possibly overdose. How do you avoid that?
Well, you have to choose materials that don't cause accumulation. So this is a big problem at the moment in the pharmaceutical industry with just poorly soluble inhaled compounds. You get lung accumulation of poorly soluble drugs, and that has to be mediated by changing the solubility profile, maybe changing the particle size to dissolve more quickly. We were interested specifically with nanomedicines to try to determine what the clearance half rate is so that you could actually estimate a true dosing frequency. I think if you tried to dose albumin on a daily basis, you might start to get accumulation of levels of albumin that the body may not be able to clear. We don't know what the effects of that would be so far. So we're taking this one step at a time and looking at designing studies to address this.
One last question? If not, let's
Thank you very
thank the
much.
speaker again.